The present topic is D10, D50, D90, how to set particle size specifications. You might have heard these terminologies like D50, D10, D90 and D100. So there is a always consideration for particle size specification for pharmaceutical formulations. Particle size of the substance, that may be API or excipient, plays a very crucial role in formulation development and formulation life cycle. Particle size of the API has a great effect on safety and efficacy. If you are working on a generic formulation, then bioequivalence of the generic formulation is required to be demonstrated, and many of the times, based on the solubility of the API, the particle size plays a crucial role in bioequivalence. Welcome all dear viewers to the video for how to set the particle size specification of pharmaceutical formulations for APIs and excipients. So in this video we are going to see what are the factors to be considered while setting the PSD limits or specification, and how the particle size specification impacts the safety and efficacy. Particle size distribution, or simply you can say it as a particle size, is the critical material attribute of the drug substance or API and also for the excipients. You might have heard the term like micronized API, milled API or fine API and coarser API. Different vendors provide different particle size of the material, and different formulations require different PSD specification. The specification of D10, D50 and D90 is required to be finalized for the formulation for manufacturing. Based on to the requirement of the formulation and formulation design, the limits for D10, D50 and D90 are given. Now, shortly I will explain that what is D10, D50 and D90. D10 means 10% of the particles are smaller than the D10 limit. D50 is the average or median particle size. That means 50% of the particles are lesser than the specified limit, or you can say that 50% of the particles are above that specified limit or criteria. D90 means 90% of the particles are below the specified limit, and once again you will get the D10 that will be more than the D90 limit. The specification should be three tier for APIs which have impact onto the safety and efficacy of the formulation based on their particle size. The particle size specification must be product specific. It should be meaningful. That means you set the specification which is actually required and which is required to be maintained. Just setting the specification for a sake, it is not good, and it will have impact onto product CQS. So always try to set the specification which is meaningful. And meaningful means what? The particle size specification should drive the product performance. That is the exact meaning of meaningful specification. It should be product specific. That means if you are making a suspension for some of the API, and also you are making extended release formulation of the same API, then you cannot and should not give the same particle size specification. You should give the specification of API based on to the product requirement and product performance. The PSD specification is required to be correlated with the drug product sequence and also with the critical process parameters and critical process attributes. PSD has an impact on to the product safety and efficacy. That's why always do the PSD specification with a great science. The specification to be set based on to the good science, and you should have the practical data in your hand while you are going to set the PSD limits. The PSD, as I have mentioned, that it is a critical material attributes of the API. Mainly this video is for API PSD. We are not going to consider the excipient PSD impact, that we will discuss in separate video. The PSD of the API affects its surface area, its flow properties and handling. PSD also affects density, both it is bulk density and tap density, and also the volume. It also have an impact on to the static nature. Sometimes the API becomes static in nature, it is not having good flow and it is difficult to handle. It affects the hygroscopicity. Fine particle size API will tend to absorb more moisture compared to the coarser. Fine particle size API will have bad flow properties. Solubility and dissolution is affected by the PSD. Also, stability is impacted by the particle size distribution. Finer particle size API are more reactive and there are higher chances of impurity formation compared to coarser API. Then manufacturability or processability. The mixing behavior, uniformity of the mixing will be impacted by the particle size of the API. PSD also affects the description, physical properties of the formulation, surface area and rate of solubilization, absorption and bioavailability. Because as it is affected the solubility, it will impact on dissolution, then dissolution will impact on disintegration and absorption and bioavailability. It will also impact onto the rate of absorption, like it is affecting the rate of solubilization. It will impact Cmax and onset of time of action and Tmax. It also impact impurity level, and also sometimes the particle size reduction techniques will tend to have impact onto the polymorphism. Sometimes melting of the API may happen due to particle size reduction techniques, and also sometimes the grinding techniques or milling of the API may lead to formation of amorphous form. Solubility is impacted by the particle size. So whenever you are dealing with the particle size, always understand the solubility, that may be BCS solubility, saturation solubility and solubility of the API in the solvents. Many of the times oils are used for making the soft gelatin capsules. So solubility of that API in the oil or lipid vehicles should be understood. BCS class, 4 BCS classes are there. Out of those, BCS class 1 and 3 compounds are highly soluble, while the drugs belonging to BCS class 2 and 4 are insoluble or they are low soluble. So always you should consider the BCS class. It is not like that if your API is BCS class 1, so PSD limit is not required. It is not like that, because not only the dissolution and permeability, the PSD affects processability, manufacturability, handling, and also the other drug product quality attributes. The formulation design should be considered while setting the PSD specification. Whether your product is immediate release, delayed release, extended release, or it is a pellet type of formulation, whether it is a suspension or solution. If you are making a solution formulation, then it is not necessary to have the particle size specification, because that API is going to be in solubilized form. But still, as you need to handle the API, you need to weigh the API and you need to solubilize it. So some particle size specification should be there. And it is always good to have particle size specification for all the APIs, whether these are soluble or insoluble, and whether these are available in the solution form, solubilized form, or these are available in the insoluble form like suspensions. Then criticality of the particle size to dissolution, bioavailability, bioequivalence, onset of action should be considered. Sometimes it is observed that the products which have fast onset of action may lead to side effects. Stability of the formulation and impurity levels are also impacted by the particle size. So also you should consider the API load in the formulation, whether your formulation contains low API load, medium API load, or high API load. For example, you are making a tablet of 1 mg API and the tablet weight is 100 mg. So it is a low dose type formulation. If you are making a 50 mg tablet and its weight is 100 mg, so the API load is 50%. So it can be said that the formulation is having medium drug load. If you are making a tablet of 900 mg API with the tablet weight of 1000 mg like metformin, then you can say that the drug load is high. Also, the manufacturing process and manufacturing steps criticality should be considered. Whether you are using the techniques to dissolve the API during manufacturing or not, or you are using the API as such. Like in pellet formulations, the API is dissolved and then it is coated onto the pellets. Description of the formulation should be considered, like mainly in the suspension or emulsion type of formulations. Also, you should understand the effect of PSD on blend uniformity and content uniformity. Also, you should have the data in your hand with different particle size API batches and different manufacturing processes like direct compression, wet granulation, roller compaction, or other manufacturing processes based on to the formulation requirement. Also, when you are setting the PSD specification, the effect of different PSD API onto drug product CQA should be understood. It is always good to have data with coarser API, data with micronized API, and data with some specific API PSD. The PSD impacts safety and efficacy, as the PSD is a critical material attribute. It should be studied in depth. PSD impacts solubility and permeability. So, if you are working onto a generic formulation, or if you are working onto a new type of formulation, or hybrid formulation like 505B2 type of formulation, always have good understanding about the impact of PSD on solubility and permeability, discriminatory and biorelevant dissolution, rate of absorption, effect on Cmax, Tmax, effect on bioavailability and bioequivalence, effect of PSD on onset of action, and side effects. PSD impacts safety and efficacy of the formulation. If the PSD has an impact onto the onset of action, duration of action, or side effects, if it is affecting Cmax and Tmax, and if you are working onto a drug like narrow therapeutic index drug, then PhD becomes very critical. If you are working onto a medication which requires rapid onset of action like analgesics, if you are working onto a rescue type of medications, antihypertensives, or life-saving medications, that time PhD of the API plays a crucial role. Also, many of the times for generic formulations, the pilot bioequivalence studies are performed with different particle size API to have the in vivo data in hand so that you can go for the best suitable particle size of API. Then after selection of the particle size of API based on to the pilot bioequivalence study results, you can go for making the pivotal batches so that sometimes uh, the aim of my this slide is to give you understanding about the effect of PhD onto the bioavailability and bioequivalence. Many of the times the particle size doesn't have much impact onto dissolution, but it shows impact in vivo. That's why many of the times it is suitable to discriminate the effect of particle size on in vivo behavior based on to the pilot studies. In pilot bioequivalence studies, you can dose the formulations with different API PhD, and then you can do the screening of API PhD and select the best suitable PhD of API. Three-tier specification for PhD is must, and three tiers means you should give the three specifications for D10, D50, D90. Sometimes D100 specification is also given. The D100 means 100% of the particles are below the specified range or within the specified limits. Then average PhD and median PhD is also given. Surface area is also required to be tested for some of the APIs and for some of the formulations. Density is also a critical material attribute, and density is driven by PhD. So density is also required to be specified. Now coming to the other requirements and considerations for giving the PhD specifications. Always have a PhD trained data from the supplier. Ask your supplier to give the PhD data of number of batches so that you can understand the batch to batch variability, and then you can go for the best suitable PhD specification. Understand the PhD uniformity supplied by the supplier or vendor and understand their process capability. Understand the analysis method, its validation, method transfer, method variability at different sites and with different sampling techniques with different batches and different analysis, different analysts, different labs, and then understand the analytical instruments, their sensitivity, and their model's effect. Reproducibility of the manufacturer's API that is required to be understood. Formulation batches data to be considered with different input API PhD and similar API PhD. You should have the stability data also for the batches made with different API PhD. You should have data for dissolution. You should understand the processability. Then other requirements are PhD consent by the supplier and batch to batch variability. Whatever the specifications you are going to set for your vendor or for API manufacturer, ask them that they are able to provide that specified PhD ranges API or not. Otherwise, what will happen? You will set the specification, but your vendor will not be able to give that specified PhD material. So always do this activity in line to your vendor. PhD of reference formulation should be studied if possible or if applicable. Different techniques are there to study the PhD of API used by the reference formulation if you are working on to a generic type of formulation. Also understand the patent and IP and legal considerations if there for PhD of the API. Sometimes the particle size ranges are claimed in the patents, so you should go to the patents and learn from the patents that what PhD is claimed by the innovator or reference formulation. The PhD is claimed in the patents because it has impact onto the bioavailability, and the PhD is very specific to the API and to the formulation. That's why many of the innovator formulation companies and organizations claim the PhD of API. Also understand the polymorphism of API and the polymorphs and their solubility. If your polymorph is different, then the solubility and particle size will be different for each type of polymorph, because polymorphism of API has different solubilities, different permeabilities, and different bioavailabilities. Different polymorphs have different solubilities. So always have understanding about the polymorphism also. For your understanding of these basic understanding about the D10, D50 and D90, I have made a separate video and it is available onto Pharma Learning in Depth channel. You can go there and watch that video. See, PhD specification should be given with a scientific understanding and with the data with the batches made from different PhD APIs. Otherwise, you will give the specification which are not meaningful. For example, I have included here the specification of D10 not more than 10 microns. D50, the particle size will be in between the 30 to 60 microns, and D90 not more than 120 microns. These specifications should not be much wide and should not be much narrow. Always use API PhD trained data whenever you are setting the PhD specification. Also, always try to set the D50, and based on that D50, you set the D10 and D90 limits. Also understand the analytical variability, and based on that, you set the PhD specifications. The video got little bit lengthy, but it is very important to consider all the critical factors for PhD specification. Always have good understanding about the wide PhD specification and narrow PhD specifications. If you give too narrow specification, your analytical method will not be or may be variable and not capable to get the result within that range, then your API may fail in the initial analysis. So consider those things also. Consider the 10 to 5 to 10 micron plus minus as a limit for setting the D10, D50 and D90. Do not set the limits like not more than 5 microns, or do not set the limits which are not possible at the analysis end. So I hope this is enough for the PhD specification, and I hope you might have got good understanding out of this video. If you have any doubt any questions, you can write in the comments and I will try to give the best possible answers. Thank you for watching the video.